Picture this: two people sitting across from each other at a dimly lit restaurant. Candles, the good silverware. Someone has clearly made an effort. And somewhere between the appetizers and the main course, one of them says something that gets said in various forms in millions of conversations every year across every culture on Earth. I just want to know. Are we exclusive? Cue the internal panic, the careful word choice, the reading of facial expressions for survival data. It is, in the modern world, one of the most loaded conversations a human being can have. And what makes it fascinating, genuinely uncomfortably fascinating, is that the anxiety surrounding it suggests the answer isn't obvious. If humans were naturally hardwired, unambiguously monogamous, that conversation wouldn't be tense. You'd both already know. The question wouldn't need asking. But here's the part that most people never think about. That conversation in various prehistoric forms was probably happening long before restaurants existed, long before candles, long before silverware. Two people, somewhere around a fire in a landscape that is no longer recognizable, navigating the same fundamental question with the same biological hardware you're using right now. So, what did ancient humans actually do? When we strip away dating apps, marriage certificates, romantic comedies, diamond ring advertisements, and centuries of religious and cultural overlay, what does the evidence actually say about how early Homo sapiens structured their intimate relationships? Were they loyal to one partner? Were they not? Was it complicated? Spoiler: it was complicated. It is always complicated. That's the entire point. Before diving into evidence, it's worth establishing why this question matters beyond satisfying curiosity. The way humans structure pair bonding has cascading effects on everything: child development, resource allocation, social cohesion, conflict patterns within groups, cooperation between groups. Understanding the evolutionary roots of human mating behavior isn't academic navel gazing. It's one of the keys to understanding why human social life looks the way it does, why certain relationship tensions appear across every culture ever studied, and why the "are we exclusive" conversation is tense in the first place. Evolutionary biology approaches mating systems through a framework that asks, what reproductive strategy, over deep time, would have produced the most surviving offspring for individuals of each sex in a given environment? The answer shapes anatomy, neurology, hormones, and behavior in ways that outlast the environment that produced them, which is why understanding ancient conditions helps make sense of modern patterns that otherwise seem contradictory. And human mating behavior is full of contradictions. Humans form pair bonds, but not always permanent ones. Humans show jealousy, but also, in certain contexts, tolerance of partners' other relationships. Humans across cultures practice monogamy as a social norm. But infidelity exists in every culture that has ever been studied. Humans produce art, poetry, and music devoted entirely to romantic love, and also have divorce rates that suggest love alone isn't always enough to sustain long-term pairing. These contradictions aren't bugs. They're the fingerprints of a complex evolutionary history that didn't settle on one clean answer. Start with anatomy, because the body carries evidence of ancestral mating systems in ways that are harder to argue with than behavioral data. Testes size relative to body mass is one of the most reliable anatomical proxies for mating system in primates. The logic is straightforward. In species where females mate with multiple males, what's called a promiscuous or polygynandrous mating system, males compete not just through physical dominance, but through sperm competition. Producing more sperm, faster, increases the probability that a given male's sperm reaches an egg before a competitor's. Natural selection, in such species, favors larger testes capable of higher sperm production. In species with strict monogamy or male-dominated polygyny, where females mate with only one or very few males, sperm competition is reduced, and testes are proportionally smaller. Gorillas, which live in harem systems where a single dominant silverback monopolizes mating access to a group of females, have tiny testes relative to their enormous body size. There's essentially no sperm competition because the dominant male prevents competitors from getting anywhere near his females. Chimpanzees, which live in highly promiscuous social systems where females mate with multiple males, have enormous testes for their body size. Sperm competition is intense and constant. Humans fall squarely in the middle. Human testes size, relative to body mass, is larger than gorilla proportions, but substantially smaller than chimpanzee proportions. The anatomy suggests a mating system that was neither strictly monogamous nor fully promiscuous. Something intermediate. Penis morphology adds another data point. Human penis shape, specifically the coronal ridge, has been studied in the context of sperm competition, with research suggesting the structure may function to mechanically displace competing sperm during intercourse. This makes anatomical sense only in a context where females sometimes mated with more than one male within a short time window. It's an uncomfortable piece of evidence for strict ancestral monogamy, because bodies don't build expensive anatomical equipment for scenarios that never occurred. Sperm motility and competition-related morphology in humans is also intermediate. More sophisticated than you'd expect in a strictly monogamous species, less extreme than in documented high-competition systems. The anatomy, across multiple systems, consistently points to the same conclusion. Humans evolved in a context that included meaningful, but not unlimited sexual competition. Now, move to skeletal evidence, because fossils tell their own story. In species with strong polygyny, where a few dominant males monopolize mating with many females, males are dramatically larger than females. This is called sexual dimorphism, and it's driven by selection pressure for male-male competition. Larger males win fights, gain mating access, reproduce more, smaller males lose. Over generations, the male average size is pulled upward by this selection pressure, while female size changes less dramatically, producing large size differences between sexes. Gorilla males are roughly twice the body weight of females. This extreme dimorphism reflects the intensity of competition in their mating system. Early hominid species, particularly Australopithecus, showed significant sexual dimorphism, suggesting their mating systems involved substantial male competition and some degree of polygyny. As the fossil record progresses through the Homo lineage toward modern humans, sexual dimorphism decreases substantially. Modern humans are only mildly sexually dimorphic. Males are on average about 15 to 20% larger than females, far less extreme than in clearly polygynous species. The reduction in sexual dimorphism over evolutionary time is consistent with a shift toward more pair-bonded social organization, where male-male competition became less intensely winner-takes-all, and the distribution of mating opportunities became somewhat more equal across males. This doesn't mean ancient humans were strictly monogamous. It means the mating system was moving away from the extreme polygyny of earlier ancestors toward something more pair-bond-oriented, while retaining features suggesting competition didn't disappear entirely. Canine tooth size tells a similar story. In many primate species, males have dramatically enlarged canine teeth used in male-male competition and threat displays. As the Homo lineage evolved, male canine size reduced progressively relative to the size seen in earlier hominids and compared to our primate relatives. Reduced canines suggest reduced direct male-male physical competition, which is consistent with pair bonding becoming a more significant feature of the mating landscape. Here's where behavioral anthropology enters the picture, and things become considerably messier. Cross-cultural survey data covering hundreds of human societies show that the majority, somewhere between 70 and 80%, depending on the sample, permit polygyny as a legal or culturally sanctioned practice. A small number permit polyandry, where one woman has multiple husbands. A substantial minority practice formal monogamy as the social norm. At first glance, that data looks like strong evidence against ancestral monogamy. If most cultures permit polygyny, doesn't that suggest polygyny is the natural human default? Not quite, because socially, permitted polygyny and actually practiced polygyny are different things. In the cultures where polygyny is permitted, most men, typically the majority, still have only one wife at a time. Polygyny in practice tends to be concentrated among high status, resource rich males who can afford to provision multiple households. The average ancestral male, in a subsistence level environment with limited resource accumulation, likely couldn't sustain more than one family group effectively. So, while the social system might permit multiple partners, the practical reality for most individuals was closer to serial or sustained pair bonding. Hunter gatherer societies, which are the closest living analog to the social structures in which most of human evolutionary history played out, tend to show relatively egalitarian social structures with lower levels of resource based status inequality than agricultural or pastoralist societies. This matters because inequality in resource holding is one of the primary engines of polygyny in human societies. When one man can accumulate dramatically more resources than another, he can attract and provision multiple mates in ways the less wealthy man cannot. In more egalitarian hunter-gatherer bands, this dynamic is reduced, and pair bonding becomes more demographically widespread. The archaeological record of the shift to agriculture, which brought food surpluses, resource accumulation, property ownership, and social stratification, correlates with increases in indicators of social inequality, including mating inequality. Some researchers argue that intensive polygyny as a widespread social institution was, paradoxically, more a product of agricultural civilization than of the hunter-gatherer past it replaced. Zooming in on what the brain and hormonal systems reveal adds another layer that popular discussion frequently misses. Humans produce significant amounts of oxytocin and vasopressin, hormones deeply implicated in pair bonding, attachment, and partner preference. These same hormones, in prairie voles, are directly responsible for the formation of lasting partner bonds. Prairie voles are monogamous. Meadow voles, a closely related species that doesn't form lasting pair bonds, have different distributions of vasopressin receptors in the brain. The receptor distribution, not the hormone itself, determines whether bonding behavior follows mating, and human vasopressin receptor distributions look, in certain key brain regions, more like the monogamy-associated prairie vole pattern than the non-bonding meadow vole pattern. This is not trivial. It suggests that the human neurological substrate was shaped, at least partially, toward the formation of lasting partner attachments. The capacity for deep, sustained emotional and social bonding with a specific individual, what most people would recognize as falling in love and staying that way, appears to be a real biological feature of human neurology, not simply a cultural construction imposed on an otherwise indifferent animal. Testosterone dynamics in human males also tell a relevant story. In many polygynous species, male testosterone remains high consistently, fueling competition, aggression, and mate-seeking behavior. In pair-bonded species, male testosterone tends to drop when males are in stable partnership relationships, particularly when offspring are present. Human males show this pattern. Testosterone levels in men in committed relationships tend to be lower than in single men, and drop further in men actively involved in infant care. This hormonal flexibility, the ability for male physiology to shift modes between competition-seeking and pair bonding, depending on relationship status, is a feature of species where both strategies are part of the behavioral repertoire. By the way, if you want to go even deeper into the secret lives of ancient humans, I just launched a full course called The Secret Lives of Ancient Humans. 10 modules, 3+ hours of content covering everything we never talk about on YouTube. What they feared, how they communicated, what hunted them, and much more. Plus an exclusive PDF guide included. Link in the description. See you inside. It doesn't mean ancient men were paragons of fidelity. It means their biology was built to accommodate pair bonding as a real mode, not just as a cultural overlay on an otherwise promiscuity-optimized system. Paternal investment is one of the most important and underappreciated pieces of this puzzle. Among primates, direct paternal care of offspring is relatively uncommon. Most primate fathers provide limited direct care beyond protection of the social group. Human fathers are unusual in the degree to which they can and do invest directly in offspring, provisioning, protection, teaching, social support across years or decades of a child's development. This matters evolutionarily because human children are extraordinarily dependent for extraordinarily long periods. Human infants are born neurologically immature, more so than any other primate, because our large brains require passing through a birth canal that became narrower as bipedalism reshaped the pelvis. The result is that human newborns are effectively gestated externally for months before achieving the developmental milestones that newborns of other primate species reach at birth. And even after that initial period, full human developmental maturation takes roughly 2 decades. That is an absurdly long dependency window by any comparative standard. A human mother in a foraging environment, nursing a highly dependent infant while potentially pregnant with the next, managing foraging, food processing, and multiple children at different developmental stages simultaneously, faces an energy and labor burden that can be partially but not fully met alone. Paternal provisioning, a mate who reliably contributes food and protection, would have substantially improved the survival chances of offspring during this extended dependency window, and improved offspring survival means the genes underlying pair bonding, paternal investment, and the hormonal systems supporting both were passed forward more effectively than alternatives. This is the core evolutionary argument for why human pair bonding may have been selectively advantageous. Not just because of the pair bond itself, but because of what it enabled for offspring survival. In an environment where raising a single child to maturity required years of intensive investment, having 2 adults committed to that investment, rather than one, or a loose coalition with uncertain commitment, would have been a meaningful fitness advantage. None of this means ancient humans were uniformly perpetually faithful in the modern romantic sense. The evidence is clear that they weren't, and understanding why is as important as understanding the pair bonding argument. Extra-pair mating, what in contemporary terms would be called infidelity, appears to have been a consistent feature of human mating systems throughout evolutionary history. Genetic analyses of modern human populations find that a small but consistent percentage of children, estimates typically range from 1 to 10% depending on the population studied, were not fathered by the man presumed to be the father. This rate is lower than in species with highly promiscuous mating systems and higher than in strictly monogamous species, which is, again, consistent with an intermediate pattern. From an evolutionary standpoint, extra-pair mating confers different advantages depending on which sex is doing it. For ancestral women, mating with a high genetic quality male outside the primary pair bond, while retaining the provisioning and investment of a committed partner, could potentially optimize offspring quality while maintaining resource security. For ancestral men, extra-pair mating represents additional reproductive opportunities at relatively low parental investment cost. Both strategies carry risks: pair bond disruption, loss of partner investment, social costs within the group. But both strategies would also, under certain conditions, have produced fitness advantages. The existence of jealousy as a cross-cultural human universal makes evolutionary sense in this context. Jealousy is a behavioral adaptation that functions to monitor and protect pair bonds from disruption. If humans had been either strictly monogamous, no threat to guard against, or completely non-pair bonded, no bond to protect, jealousy as a persistent, intense emotional response wouldn't have been selected for. The fact that it's universal, and that it shows predictable sex differences in the triggers that produce the most intense response, is consistent with a mating system where pair bonds were real and valuable, but also under recurring competitive pressure. The role of social groups in ancient human mating is another dimension that the modern framing, 2 individuals, 1 relationship, tends to obscure. Early Homo sapiens lived in bands of roughly 20 to 50 individuals with broader social networks connecting multiple bands. Mating wasn't a purely private bilateral negotiation. It occurred within and between social groups, with implications for alliances, resource sharing, and intergroup relationships. Some researchers argue that pair bonding in humans served not just the reproductive interests of the 2 individuals involved, but also functioned as a social institution that reduced within-group male-male competition. A pair bond norm that distributed mating opportunities more broadly across males reduced the destabilizing effects of intense competition for the few females in a small band. This is sometimes called the male cooperation hypothesis for pair bonding. That widespread pair bonding was selected for partly because it reduced within group conflict and enabled the sustained male cooperation that made human group hunting, defense, and resource sharing strategies possible. A band in which a few dominant males monopolized all female access, guerrilla style, would be one with a large number of reproductively excluded males with limited incentive to cooperate with the dominant males who were out-competing them. A band in which most males had committed pair bonds had a different internal dynamic. More mutual investment, more stable coalition formation, more effective collective behavior. This doesn't mean the social norm was perfectly adhered to. Social norms rarely are, but the existence of a pair bond norm within the group, even imperfectly followed, may have produced better collective outcomes than the alternative, which would explain why pair bonding as a social institution became widespread and persistent. Women's mate preferences, which evolutionary psychology has studied extensively, reveal something important about what ancestral environments we're selecting for. Across cultures and contexts, research consistently finds that women weight indicators of resource acquisition capability, social status, reliability, and commitment more heavily in long-term partner selection than in short-term contexts. This preference pattern is consistent with a history in which long-term paternal investment was a meaningful variable in offspring survival. If it hadn't mattered, the preference for it wouldn't have been selected for. Women who, ancestrally, were attracted to and chose partners likely to invest in offspring would have raised children with better survival odds than women who selected partners on other criteria alone. Men's mate preferences show a different pattern. More emphasis on physical indicators of fertility and reproductive value in mate selection, with somewhat different calculations depending on whether the context signals a short-term or long-term interaction. This is consistent with a mating system where males faced both pair bond contexts and extra pair opportunity contexts, and the preference systems shifted somewhat between them. Neither of these preference patterns is deterministic. Their tendencies built on ancient statistical regularities, operating in a modern environment that is vastly different from the one that shaped them. A preference that made adaptive sense in a small foraging band doesn't map cleanly onto modern dating in a city of millions, which is part of why modern mating produces the particular combination of intense longing, confusing behavior, and inexplicable attraction that it does. Zoological comparison brings one final useful perspective. Humans are sometimes compared to gibbons, which are commonly cited as one of the few primate monogamous species. But the gibbon comparison is instructive in unexpected ways. Gibbon monogamy is somewhat more complicated than its reputation. Extra pair mating occurs in gibbon populations. Pair bonds sometimes dissolve and reform with new partners, and the social reality doesn't perfectly match the textbook description. More importantly, gibbons don't have pair bonding enforced by neurological reward systems the way prairie voles do. Their pair bonding appears to be more circumstantially maintained and less emotionally intense than what humans describe in the experience of romantic love. Human pair bonding has a neurological intensity and an emotional complexity. The obsessive early phase of romantic attachment, the deep security of long-term bonded partnership, the devastating pain of bond disruption. That suggests something more robustly wired in than simple circumstantial pairing. The neurological intensity of human romantic attachment is difficult to explain if pair bonding were simply a cultural overlay on an otherwise indifferent biological substrate. Brains don't generate that level of emotional investment, that degree of neural reward activation during early attachment, for systems that don't matter. The pain of heartbreaks, which involves some of the same neural pathways as physical pain, is the immune system of the pair bond. It hurts that much because the bond was supposed to matter that much. So what's the answer? What did ancient humans actually do? The evidence, taken together, paints a picture of a species that was, and remains, what researchers sometimes call socially monogamous, but not sexually monogamous. Pair bonding was real, biologically supported, and likely adaptive. Most individuals, most of the time, formed primary partner attachments that involved sustained cooperation, shared investment in offspring, and genuine emotional attachment. These bonds were not just cultural inventions imposed on an indifferent animal. They were supported by hormonal systems, neurological reward architecture, and preferential mate selection patterns that go deep into the biological substrate. At the same time, strict lifelong sexual exclusivity was probably not the statistical norm. Extra pair mating occurred. Pair bonds dissolved and reformed with new partners. The capacity for both deep pair attachment and attraction outside that attachment coexisted in the same individuals, which is exactly what you would expect from a species whose anatomy, sperm biology, and behavioral repertoire all point toward an intermediate mating system. Ancient humans, in other words, had the same basic problem that modern humans have. The pair bond is real. The attachment is real. The value of sustained partnership is real. And so are the competing impulses that test it. The tension wasn't invented by civilization. The cultural and legal frameworks around marriage, fidelity, and commitment were developed in large part as social technology for managing a tension that was already there in the biology, which means that couple at the restaurant, sitting across from each other in candlelight, navigating the loaded question of exclusivity, they're not dealing with a modern problem. They're dealing with a very, very old one, using the same neurological hardware, the same hormonal architecture, the same emotional intensity, and complicated competing impulses that their ancestors were navigating tens of thousands of years ago around a fire. The context changed. The question didn't. And if it's any comfort at all, which it might not be depending on how dinner is going of Rich Fravorosaurus, the fact that the conversation is tense is not evidence of personal failure or civilizational dysfunction. It's evidence that you're a human being, running ancient software in a modern world, trying to work out something that evolution deliberately left ambiguous, because evolution doesn't optimize for simplicity, it optimizes for survival. And apparently the complicated version of human pair bonding was complicated enough and functional enough to get us all the way here.
Somewhere in your body right now is a chemical signature left behind by an ancestor who was never faithful, not out of choice, out of biology. Most people assume monogamy is humanity's natural state. One partner, one bond, hardwired from the beginning. The evidence tells a messier story. Biologists studying mammals have found a reliable pattern. Truly monogamous species tend to show almost no physical difference between males and females. Humans don't fit that pattern. Men are, on average, notably larger than women, a trait biologists call sexual dimorphism, and one strongly associated across species with competition for multiple mates, not lifelong pairing. Genetic studies push the picture further. Research comparing human Y chromosome and mitochondrial DNA diversity has found evidence of a striking historical imbalance, suggesting that for long stretches of human history, a smaller number of men fathered a disproportionate share of children. That pattern doesn't emerge from strict universal monogamy. Anthropological surveys of documented human societies tell a similar story. A majority have historically permitted polygamy in some form. Strict lifelong monogamy as a cultural rule appears to be far less universal than most people assume. None of this means ancient humans lived without bonds. Pair bonding, long, close attachments between partners, does appear consistently across human history, likely shaped by the demands of raising children who need years of care to survive. But biological pair bonding and strict sexual exclusivity are not the same thing. Humans, the evidence suggests, evolved for connection, not necessarily for faithfulness. We tend to treat loyalty as a moral test our ancestors either passed or failed. The science suggests something less judgmental, a species built for deep attachment, operating inside a body that was never fully built for exclusivity, and spending thousands of years negotiating the gap between the two.